Hi students, in this video presentation I'm going to talk about my favourite developmental model, which is the chicken embryo. So my lab uses the chicken embryo as a developmental model to study two important processes. One is sex determination, so that's the differentiation of testes versus ovaries during embryonic life, and the other is limb morphogenesis. Now, we've touched on the chicken model earlier in the unit, and in these next two prac classes we're going to give you some hands-on experience with this model. So there's a video on Moodle where I talk about the advantages of using the chicken model. I'll just summarise them again briefly here. There are three main advantages. Firstly, the chicken is the closest non-mammalian model to humans, and so this means that developmental processes are conserved across the two species. And so the chicken is not too close to human but not too distant. And so this means that if we're looking for conserved regulatory elements, for example, in the genome, we can pinpoint them by comparing human and chicken. This is a bit easier to do than comparing, say, human and mouse, where the genomes are more similar, and so it's hard to tease out those conserved regulatory elements. A second important advantage of using the chicken is the fact that development occurs in the egg outside the maternal body, and so this makes it very accessible to experimental manipulation. We can open an early developing embryo, open an egg, access the embryo. We can inject DNA constructs or knockdown constructs into that embryo, seal the egg again, place it back in the egg incubator, and then open it again, say a few days or a week later, and look at the effects that that manipulation has had. And so this is a more rapid and a more cost-effective method than, say, mouse transgenics. So lastly, the chicken embryo is quite useful because of its large size. And so if you compare the same developmental stage in a chicken and mouse, the chicken is larger, and so this means that we can often obtain larger amounts of tissue for downstream genetic or biochemical analysis, so getting more RNA or getting more protein, for example. So in this prac, we're going to look at ways of opening the chicken embryo and manipulating embryonic development. As I've said, our group works on sex determination, where we look at the embryonic gonads by injecting DNA constructs or knockdown constructs. This is a bit of an art, so instead we're going to look at a more tractable system, an easier tissue to access, and that's the developing limb. So the limb bud obviously develops externally, and it can be readily manipulated in the chicken model. The developing limb buds of the chicken embryo become, in the case of the forelimb buds, the wings, and the hind limb buds become the legs. And so there are two important signalling centres in the early forming limb bud. The apical ectodermal ridge, or the AER, and this is a specialised area of ectoderm around the edge of the limb bud which coordinates outgrowth and proximal distal patterning. And the other important signalling centre is the zone of polarising activity, or ZPA. This is a specialised area of mesoderm or mesenchyme in the posterior part of the limb bud, and it patterns the limb bud across the anterior posterior axis, so from the thumb across to the little finger, for example. So the aim of this prac today is to disturb the ZPA, that posterior piece of mesoderm in the developing hind limb or the forelimb bud, and then look at the effects that this has on limb morphogenesis. This figure here shows you embryonic development in the chicken. And development is staged according to various morphological criteria that Hamburger and Hamilton in the 1950s devised. And so they developed a series of embryonic stages, as shown here. This was published in the 1950s, and the paper was one of the most, or has become, one of the most widely cited papers in developmental biology, because people working on chicken embryos are always quoting the developmental stages. And so we today are going to be looking at manipulating the limb buds of day 3.5 embryos, and this corresponds to developmental stages 21 to 22, highlighted here. So we will open eggs at day 3.5, manipulate the ZPA, that posterior mesoderm, in the limb buds of either the forelimb or the hind limb bud, and then we'll reseal the egg and allow development to proceed for another week, and then we will look at the phenotype of the limb 1 week later. So we're going to open an egg at day 3.5, and there should be 1 egg per student, and then gently tear away a part of the ZPA in the posterior pole of the limb bud, then reseal the egg for 1 week, as I mentioned. And now I'll show you how to do this. So firstly, take the egg and place it in a holder like this, and then we need to pierce a hole in the egg. Bracing the egg with one hand, tap a hole in the blunt end of the egg. Here. Using a 3mm syringe and needle and angling it downwards, gently insert the needle and draw out 3 mL of albumin. Next, bracing the egg, tap another hole at the top of the egg here, as shown, with forceps, and gently peel away some eggshell. And so the embryo now drops below the top of the eggshell as air rushes in because you've removed 3 mL of albumin out the side. And using scissors, cut a larger hole so that you can access the embryo. When you place the egg under the microscope, you can see the embryo as shown here. This is a day 3.5 to 4 embryo, and you can see the heart clearly beating in the head at the top of the image and the 2 limb buds. Now what we want to do is to manipulate either the forelimb or the hind limb bud, and you can choose which one you'd like to do. There is a clear vitelline membrane over that embryo, so you want to try and pull that away first. And I'll show you how to try and do that with these forceps. Now it can't be seen, that vitelline membrane, but I've torn it away. Now the aim is to snip away a part of the posterior pole of the limb bud, so this is ZPA. And I'm going to try and do that now on the forelimb bud. This may require higher magnification, so I'm going to zoom in on the embryo. This is the forelimb bud. And I've just snipped a portion of the posterior pole away from the limb bud. So I will zoom out. And you can see there that that's been damaged or disturbed. The aim now is to seal the egg, return it to the incubator, and observe the effects upon the development of the limb bud now that we've disturbed part of the ZPA in the posterior pole of that limb bud.